Hello from Australia. I hope you're enjoying this wonderful online global forum. I'll also be talking about the beautiful Richmond Birdwing. However, the focus of my presentation is how we are addressing one of the key threatening processes the butterfly is facing here in this country. I'll describe a state government project I've been involved with in Queensland that uses captive breeding and release of Richmond birdwings to overcome inbreeding issues in wild populations. This work really represents just one component of overall recovery efforts for the species in this state. Now, the work is really a partnership between my department, the Queensland Government's Department of Environment and Science, and in fact, two different arms within that department, the Threatened Species section and David Fleay Wildlife Park on the Gold Coast, where the main captive breeding facilities are. It also involves the Richmond Birdwing Conservation Network, which is an affiliation of individuals, groups, and organisations dedicated to the conservation of the Richmond birdwing and its two native larval food plants. And the third partner is Dr. Don Sands, who was working with CSIRO originally when this project began and has since retired. He has been foundational to the whole project, as I'll explain later. I'll just move that box out of the way. Now, if you concentrate on the map at the top right, it shows Australia, and the heavy outline rectangle shows where Queensland is in that overall map of the country. And here we have three species of birdwings. We have the New Guinea birdwing in the far north, shown in the red, the Cairns birdwing in the wet tropics, another population just to the south of that, and the purple at the very bottom on the right shows the Richmond birdwing's distribution, straddling the Queensland and New South Wales border. So in fact, the Richmond birdwing is Australia's only subtropical birdwing species. And although it's a large butterfly, it's characteristically the smallest of the three that we have here in this country. Now, if you concentrate on that map area at the bottom right, I'll look at that in a little bit more detail. This map shows both historical and more current distributions of the species. If you concentrate on the yellow dots to start with, the very north at River Heads near Fraser Island, that was the northernmost extent of known historical records for the species, and it stretched as far south as Grafton in New South Wales, which was a north-south distance of about 470 kilometres. Now, if you concentrate on the green dots, you'll see that they occupy a much more restricted area. There's been quite significant range contractions from the both north and south of the species distribution, and there's been local extinctions in the middle, particularly around Brisbane, which you can see to the left of my speaking box. And that's the centre, the urban centre for Queensland, the capital, and the species is no longer breeding in this area. So what has in fact happened is that we've ended up with two distinct populations, north and south of Brisbane. So it is a species under threat. And because of those range contractions and decreases in abundance, it has been listed as a vulnerable species in Queensland under state legislation, although it's not listed in New South Wales or in fact nationally as yet. Now, the threatening processes that led to that listing in Queensland are the usual culprits, things like habitat destruction and fragmentation, inbreeding depression, which I'll focus on more in this talk, climate change through both effects on increasing temperatures and drought effects affecting the butterfly and its food plants. Invasive weeds are a particular problem, particularly in rainforest corridors along riparian areas. And one particularly bad invasive weed is Dutchman's pipe, a South American species that is invaded. It belongs to the same family as the butterfly's natural larval food plants, but in fact it is toxic. So when the adults lay eggs on it and the larvae begin feeding, they begin to die. But the main focus of my talk is inbreeding depression, which is linked to habitat fragmentation, as I'll explain. So habitat fragmentation is particularly insidious because it causes these isolated pockets of habitat with reduced opportunities for butterflies or other species, in fact, to move between them. And that lack of genetic exchange between these isolates leads to inbreeding depression of local populations. And in fact, an example of that is seen where there's a red arrow points. This is Burleigh Head National Park, on the Gold Coast of Queensland, where a population of the Richmond Birdwing and its host vines still occurs. And inbreeding depression has been noted in that particular population. So what exactly is inbreeding depression? Well, as I've said, it occurs where individuals are isolated and they don't have much opportunity for exchanging genetic material between neighboring populations. And usually the numbers of individuals are quite low. And it manifests itself in a whole range of ways through egg mortality, through protracted larval development, and sometimes increased mortality of larvae. And sometimes the pupae fail to emerge, so they just die before the adult stage. And if adults do emerge, they're usually smaller and lack vigor and fitness. And if they are able to breed, they don't lay as many eggs. So those sorts of effects on the various life stages of the butterfly were first observed in captivity by Dr. Bert Orr in Australia with work he did on several papilionid butterflies, including the Richmond Birdwing. But as I've mentioned, they've also been seen in wild populations, and if they're left to run wild and unchecked in those wild populations, it can lead to local extinctions or extirpations of those local spots. So how can we overcome inbreeding depression? Well, indirectly, you can begin restoring habitat connections between those isolated fragments. Or you can at least put stepping stones of larval food plants for the adult butterflies to lay on in between those. So you create these tenuous links between the existing habitat chunks. Now, all that takes quite a bit of time. So a more direct action is to actually translocate individuals between the habitat fragments. Or another alternative is to selectively mate individuals from separate populations so that you're actually achieving outbreeding and then release the progeny of those matings back into the wild in the key locations. And that's actually the approach that we're adopting in the project I'm describing today. So the aims of the project that I'll talk about is to use selective mating and captive rearing to overcome inbreeding depression. And we do that by mating unrelated adults, getting the females to lay eggs in captivity, and then rearing the resulting offspring. And then we can take those back out into the wild and release them, and thereby get reinvigorated genetic mix in the wild population, and therefore improve conservation prospects for the species. So the idea and initial impetus for all of this work was provided by Dr. Don Sands. He and members of the Richmond Birdwing Conservation Network were responsible for doing the initial experiments to show that this approach was in fact feasible. And he then encouraged us to take it up and in fact expand it as much as we could. So it's all Don's fault, in other words. So the first thing to do for starting captive breeding is to source founder stock. So the map on the left shows the southeast Queensland area with little tiny yellow triangles showing where we've sourced founders from. You probably can't read the details on the right, but the basic information there is that we have sourced founders from a great many locations. The main thrust of the approach, though, is to ensure that the founders come from different genetic stock. And the way we're achieving that is based on the life history of the female butterflies. We know that they can fly at least 30 kilometers from their breeding area. And so we've been trying to ensure that we take founder stock, males and females, from at least 40 kilometers physical separation, so that we have more chance that they are of different, have different genes represented in the captive population. So in the captive facilities, we have to establish the vines that the species lays on and the larval stages feed on. And this is the lowland birdwing butterfly vine. It's called Pararistolochia pravenosa. And on the bottom left, you can see a photograph of a whole lot of vines that were acquired because of a funding grant given to us by the Swallowtail and Birdwing Butterfly Trust. They were very kind and generous in providing £500 back in 2018 for us to acquire more vines. So we also have to manage the birdwings in these captive facilities. And on the right-hand side, you can see one of the facilities we've got at David Flay Wildlife Park. It's a flight enclosure, but the larvae are able to free roam in there and move between vines as they like. So a major part of managing them in these captive situations is to be on the lookout for predators like spiders and geckos, and be sure that males and females that you don't want to mate are not mating. So genetic management is a big part of that. It was also became very obvious to us quite early that we needed a second facility to help manage genetic stocks and keep them separate. And because this one is not predator proof, we have to use rearing sleeves to keep the larvae safe and the pupae safe while they're in this facility. Now, captive husbandry, the founders usually involve collecting eggs and rearing those, or getting them to hatch the larvae and getting the larvae through to pupal stage. And then once the pupae hatch, we have the adults, the precious adults for the mating, because that's the key to success of the project. So keeping the adults healthy means feeding them nectar. Some of the adults are immediately able to feed themselves on potted plants or cut blossom from the wild, but very often we have to give them artificial nectar solutions, either by introducing them by hand, but through various means we can get them to feed themselves, like on the lower right photo. They're very highly attracted to bright colors and come and feed on the nectar in amongst those beads. Once we have males and females that we want to mate, we did initially start mating them, or letting them choose their own mates by putting the males and females together and waiting for them to do what they need to do. But it became obvious to us that it was much more efficient and we could get the pairings that we really wanted by doing hand mating, which is shown here at David Flay Wildlife Park. So that's been the key to success of getting the female and male pairings that we're after. So those females then lay eggs in captivity, and it's that outbred progeny, that first generation, the ones that we wish to release to the wild to increase the genetic diversity back in those wild populations. So the idea is that once we've reared them to mid-stage or late-stage larvae, we transfer those to selected sites and release them, usually in batches of 30 or so, so they can get a decent population established. And then we go back over several years and do subsequent releases to maintain the population at that site and keep the diversity in the gene in that population. So once we release the larvae in the wild, they generally crawl off and start feeding for themselves. So they adapt very quickly to this situation. Now the results that we've had to date, up till about 2018, is shown on the map on the left. Well, this is really where the translocations have happened in southeast Queensland. The size of the orange blobs shows a number of individuals that have been translocated there. And we've done translocations that represent reintroductions where the species was actually had become locally extinct at that site, and supplementations where the numbers were dwindling and showing signs of inbreeding, and we've supplemented them with some outcross material. Just from a statistics point of view, we've released just under 500 individuals, outbred individuals, across 10 sites in southeast Queensland in that period since the project began. Now, if I tried to explain what I think the impact of that has been on wild populations, the map on the right shows yellow blobs that represent breeding distributions at the start of this project for the species. So there were a couple of separated populations to the north of Brisbane, and one from the Gold Coast stretching south into New South Wales to the south of Brisbane. The orange blobs show where we've done releases. So at the top right there, you can see one labeled Catharaba. So this is a site where the species had disappeared since the early 2000s and had not been seen in the intervening period. But after our program of reintroductions, the butterflies were again observed flying at that site. And so the lost population was again flying and and thrilling the local landholders in that area. And what's more is that the adults and then larvae were seen at distances of up to 20 kilometers from those initial release sites. And so the great thing about that is that the species appears to be not only re-establishing the local area, but also joining up with the existing wild populations further to the south and southwest. The same thing was achieved at Coolum, and the butterflies are still being seen there. And monitoring is showing that natural breeding is continuing in these sites more than 10 years after the initial releases. So it's been an exciting development. In the south of the range, the releases we've done have really been supplementations to areas where the butterflies were present, but in low numbers. But anecdotally, reports from members of the public in that area suggest that there's been a tremendous increase in birdwing abundance in the areas surrounding those locations. So that's very heartening as well, although not definitive proof. So currently the project is on pause because we're awaiting a refurbishment of a new breeding facility at David Flay Wildlife Park. That's it in its preliminary stages. This has got multiple components, compartments within it that allow us to ensure that stock of different genetic origin can be housed separately, or we can keep males and females separate and only mix them when we want them to breed. And when operational, this facility should really let us achieve a much bigger productivity in producing our bred progeny for release. So the next steps really are to get that program reinvigorated and underway again using those refurbished facilities. We want to get founder stock from new wild populations, so those with some different genetics than those we've sourced to date, and then continue the outbreeding efforts and targeting key sites to boost the genetic diversity of the species in the wild, and follow that up with annual monitoring to check that the species is hanging on where we've done these releases. But a major part of the work is really to encourage and support the Richmond Birdwing Conservation Network, local governments, conservation groups, and just members of the public who are continuing to plant vines to expand the range and abundance of food resources so that the birdwing can actually naturally reconnect its populations, its isolated fragments. So the aim of the captive breeding was really a stopgap, short-term measure because there was alarming contractions in range. But the long-term success of the project will really depend on this habitat and food plant restoration project. So the two working arm in arm should really achieve positive things for conservation of the species. So a project like this doesn't happen without the help of a lot of people, and just some of those people are shown in this slide. It has been a tremendous effort from huge numbers of people across southeast Queensland and in northeast New South Wales. And should you need to get in contact with me, here are my details. Thank you very much.